Namaskar. Today we'll study autophagy. Autophagy is a highly conserved degradation pathway for bulk cellular components, and the cells actually here the cells. So this occurs inside the cell, and the cells degrade the dysfunctional and unnecessary cellular components. The organelles are degraded by the cell itself by the process of autophagy. We can call them as the cargo, the proteins and the impaired organelles which are dysfunctional, unnecessary. They can be removed, degraded by this process of autophagy. And so we can say that it is a process which promotes the survival of the cell during the starvation period. Say during the starvation period there is not enough nutrient for the cell, so the cell will develop this mechanism of autophagy so that the cellular energy level will be maintained and the unnecessary dysfunctional components will be removed so that the cell can survive. However, excessive autophagy will lead to cell death. Excessive autophagy will lead to cell death. So what happens in autophagy? The impaired organelles and unwanted cellular components, they first form the autophagosome. Then that autophagosome will fuse with the lysosome present in the cell itself. Okay. And so this is cell, this is nucleus, this is lysosome. So there will be first formation of autophagosome, then it will fuse with the lysosome, and the degradation of all those unnecessary components or cargo by the lysosomal enzymes, lysosomal hydrolases. Let us see here. So first there will be one isolation membrane to which these cargo, the unwanted organelles, unnecessary proteins, they will adhere, and that will result in the formation of a double membrane enclosed vacuole, and so there will be massive autophagic vacuolation of the cytoplasm, and we call it as autophagosome, and this occurs in presence of class 3 phosphoneinositide 3 kinase, class 3 PI3K, autophagy related gene 6 ATG6, ubiquitin or ubiquitin like modifications of the target proteins. Now this autophagosome will fuse with the lysosome of the same cell. So some of these cytoplasmic components form this autophagosome, so that is we call as autophagic vacuolization. Then this autophagosome will fuse with the lysosome, so the lysosomal hydrolases, the enzymes, will degrade these components of autophagosome. We call this complex as the autolysosome, and there will be autophagy, and some of the cellular contents will be degraded in the cell. So this is all about autophagy, and how this differs from necrosis and apoptosis we'll discuss. So how this autophagy this autophagy occurs in certain physiological conditions like cell survival, cell metabolism, development, and aging, and during pathological conditions like cell death, infection, immunity, cancer, neurodegenerative diseases, metabolic diseases, ischemia, reperfusion, injury, this autophagy is associated with all these physiological and pathological processes. And how this differs from necrosis? Necrosis is cell death, and autophagy we call as anti-necrosis. So cell survival. The cell wants to survive, it will undergo autophagy. This by necrosis there will be cell death. And what is the difference between autophagy and apoptosis? In autophagy and apoptosis difference, mainly the source of lysosomal enzymes. In apoptosis, what happens? The lysosomal enzymes of the phagocytic during the phagocytosis, the blebs generated by the see, this is apoptosis where the cell shrinkage occurs, the formation of blebs occur, and these blebs show phosphatidylserine on their surface, express phosphatidylserine, which will attract the macrophages for phagocytosis, and they are phagocytosed by the lysosomal enzymes. So the phagocytic cell lysosomes actually cause the phagocytosis of blebs in case of apoptosis, whereas the endogenous lysosomal machinery of the dying cell itself cause the degradation of unnecessary cellular components in autophagy. This is the difference between autophagy and apoptosis. Thank you.